Hello everyone, this is Vaishnavi Swetha studying BPHARM second year in Maharaja's College of Pharmacy. Today my seminar topic is acid fast staining. Acid fast staining is a very important topic in microbiology which helps to differentiate acid fast bacteria from non-acid fast bacteria. Coming to the principle of acid fast staining, acid fast bacteria has large amount of lipids and mycolic acid in its capsule, whereas non-acid fast bacteria lacks it. These differences made them to react differently in acid-fast staining process. We already know that the bacteria cell wall is in turn covered by a hard shell known as capsule. So based on the composition of the capsule in acid-fast and non-acid-fast bacteria, that is, the acid-fast bacteria will have mycolic acid in its capsule, whereas the non-acid-fast bacteria will not have the mycolic acid in its capsule. So because of this difference, they react differently in the acid-fast staining process. We already differentiated the gram-positive bacteria from gram-negative bacteria by gram staining. So again, the gram-positive bacteria are divided into acid-fast bacteria and non-acid-fast bacteria. To difference to differentiate the both acid-fast and non-acid-fast bacteria, we will follow this process. The main requirements are carbamazepine, which acts as primary stain, heat, which acts as mordant, acid alcohol acts as decolorizing agent, methylene blue acts as secondary stain. The method we follow is, first we will apply the primary stain carbamazepine to the sample having both acid fast and non-acid fast bacteria. This is the primary stain carbamazepine which will impart red or pink color. To the sample containing acid fast and non-acid fast bacteria. Later, we will heat fix the slide using the flame. Here, the heat fixation is indirectly acting as a mordant. Mordant is an agent which will fix the stain to the bacteria. So here, heating acts as a mordant. Next, we will add acid alcohol. Here, acid alcohol acts as decolorizing agent. So, as the acid alcohol is added to the sample, the acid fast bacteria will not lose its carbamazepine stain, whereas the non-acid fast bacteria will lose the carbamazepine stain. Why? Because the presence of mycolic acid in the acid fast bacteria, the acid fast bacteria will retains the carbamazepine stain and will remain in the pink or red color even after the addition of acid alcohol. Whereas in the non-acid fast bacteria, there will be no presence of mycolic acid in its capsule. So after the addition of acid alcohol, it will lose its carbamazepine stain, that is pink or red color, and it will becomes colorless. So later we will add the secondary stain methylene blue, and after the addition of methylene blue, the acid fast bacteria will not absorb the methylene blue as it already present in the pink or red stain. But the colorless bacteria, that is non-acid fast bacteria, which is present in colorless after the addition of acid alcohol, will absorb the methylene blue color and will becomes blue. So in the final conclusion, after we observe the sample under the microscope, we can observe the acid fast bacteria appears as a red or pink color as it retains the carbamazepine stain up to the end of the process. But the non-acid fast bacteria will absorb methylene blue and observed as blue color under the microscope. So the conclusion is the primary dye application, that is the carbamazepine reagent to the sample, will shows both acid fast and non-acid fast bacteria in the red or pink color. After the addition of decolorized acid alcohol, the acid fast bacteria will remains as red in color, whereas the non-acid fast bacteria will becomes colorless. After the addition of counter stain, that is methylene blue, the acid fast bacteria will remains as red in color, whereas the non-acid fast bacteria will becomes blue color. So based on the differences in the composition of their capsule, they react differently with the stains and the decolorizing agents we added, and they will show the different results at the end of acid fast staining process. So based on this observation, we can easily differentiate the acid fast from the from non-acid fast bacteria. Thank you.